Hello everyone. So this is the video lecture for meiosis. And in this video lecture, we're also going to cover the concept of sexual reproduction. So here we go. So meiosis is yet another form of sexual reproduction. So let's make a note of that. And it is all based on the movement and interaction and distribution of homologous chromosome pairs. So if you think back to the way human chromosomes work, we have 46 chromosomes total. So if we broke those down into pairs, there would be 23 pairs of chromosomes. So literally 2 copies of each kind of chromosome. Why do we have 2 copies? Well, you are a fusion of your parents. Half of your chromosomes come from your father, the other half come from your mother, and when they come together, they produce a zygote, which turns into a fetus, and that fetus eventually turns into a baby. So the thing is, in meiosis, we have the condition or the situation where we have to take the 46 chromosomes that all human cells have and somehow narrow that down into just half the total amount. So right away, that's one of the main ways that meiosis is going to be different from mitosis, because meiosis is going to produce 4 daughter cells, but those daughter cells are going to be genetically different. They're only going to have half the parent chromosomes, okay? And this is the basis of genetic variation. Because I'm making 4 different cells, and any of those cells can be fertilized with a sperm cell, fertilization where a sperm cell and an egg cell come together, that could be any combination, then I'm going to have differences. If you think about your family, even if you and your siblings come from the same mom and dad, y'all are not carbon copies of each other, and you are not carbon copies of of either parent either. You are a combination in different amounts, okay? So sexual reproduction is going to produce offspring, babies, by uniting a male sex cell and a female sex cell. We also call those sex cells gametes. And in common lingo, we refer to them as sperm cells and egg cells. So another word for gametes are sex cells. And we like to differentiate because when we do mitosis, where we're making exact replicas of the original cell, that's how we make somatic cells or body cells, okay? So meiosis is going to produce gametes with half the chromosome number, whatever that chromosome total is for that individual organism. Meiosis is going to cut that number in half. In the case of humans, we go from 46 chromosomes to 23 chromosomes. So because we make 2 different sets of sex cells, males make sperm and females make eggs, the process of making sperm cells is called spermatogenesis. It occurs in the testes in males. And for females, the production of what will eventually become an egg cell is called oogenesis. It's where we make follicles, and eventually those follicles mature into eggs, and that happens in the ovaries. So to get the 2 together, to combine a sperm cell and an egg cell, the process that ensues is called fertilization. So it's the fusion of the nucleus of the sperm cell and the egg cell. So this is how we go back to having our total number. So in human cells, that would be 23 plus 23, 23 in the sperm, 23 in the egg, and that's how we go back to having our 46. So when the 2 nuclei of the egg cell and the sperm cell fuse together, what they produce is called a zygote cell. And that zygote is the result of fertilization. So now that zygote cell is going to go through lots and lots of cell division to go from 1 cell into billions of cells. So this picture shows the process of meiosis as it occurs to make sperm cells and then to make egg cells. So something to note, they all look pretty similar in terms of how we start off. And then we go through meiosis 1. So something I need to mention is meiosis is a double division, which is how we're going to end up with 4 cells instead of just 2 cells. So after meiosis 1, we've made 2 cells in both sets. And then meiosis 2 takes place, and each of these original cells from meiosis 1 will make 2 cells. So we'll have 1, 2, 3, 4 cells here, and then 1, 2, 3, 4 cells there. So I've already referenced quite a few of these terms, but just make sure you understand the difference between somatic cells. Remember, somatic cells are all your body cells except for the gametes, and they are made or produced through mitosis. Of course, your gamete cells are going to be your sperm cells or egg cells, depending on if you're male or female biologically, and they are produced through meiosis. Autosomes are the names that we give to the chromosomes 1 through 22. And these chromosomes are going to contain the majority of your genes, thousands of genes. The last pair, pair 23, is known as the sex chromosomes. And they contain hundreds of genes. And specifically, they're the ones that are going to have different genes based on whether you're male or female. So human females have an XX chromosome 23, and males have XY chromosome 23s. XX is fully homologous. They're exactly the same. XY is not fully homologous because Y has different genes than X. Okay, so the resulting gametes, a female can make 2 X gametes or 2 X cells, and a male can make 1 X cell and 1 Y cell. So this is an example of a human set of chromosomes. And what they've done is, this is kind of what it would look like in the nucleus. The chromosomes are just all over the place. There's no real rhyme or reason. But we can also pair the chromosomes up and find chromosomes 1 all the way through chromosomes 23 based on size and shape. So here we have our 23 pairs of chromosomes. Remember, from chromosome 1 all the way through to chromosome 22, those are going to be the somatic chromosomes. And then lastly, the pair 23 is the gametic chromosomes, so the determination of biological gender. So this picture is known as a karyotype, and it's one of the ways that we can look at chromosomes to see if there are deformities or if we have extras or if we're missing some or anything along those lines. All right, so if we were to get a bird's eye view of what's going on inside of our nucleus, so here's our cell, here's our nucleus, and now we're looking in the nucleus. Here I can see the nucleus with some chromosomes depicted. If I could zoom in on one of those chromosomes up close, this is kind of what it looks like, you'd notice this banding pattern. That banding pattern showcases how the genes are layered on that chromosome. So if I could zoom in on a section of those bands, then I would see DNA. And then if I got even more up close on the DNA, I'd be able to see the actual material that makes up our DNA. So our sugar phosphate backbone, and then our nitrogen base is made up of A's, T's, C's, and G's. Okay, so let's talk about homologous chromosomes again. So homologous chromosomes are the chromosomes you inherit from your parents. So sometimes we call them parental chromosomes. And you get them from your male parent, so paternal chromosomes, and you get them from your female parent, maternal chromosomes. They are homologous because they carry very, very similar information to each other. Now, just because the chromosomes come in pairs doesn't mean that they are going to carry exactly the same information. So we use the term alleles, and an allele is a version or type of gene. Okay, so a version or type of gene. So for example, you might have a gene for curly hair versus a gene for straight hair. Okay, so maybe your dad has a curly hair gene, maybe your mom has a curly hair gene. They're each going to give you a gene for hair texture. It just depends genetically. What they have is what they will pass on. Okay, so in the process of meiosis, those homolog pairs do separate. Before meiosis, those cells that are about to go through meiosis are going to be diploid. They have the total chromosome number. So in the case of humans, that would be 46 chromosomes. But after meiosis, every single one of the cells that meiosis produces is now going to be haploid. We're only going to have 23 chromosomes. So before meiosis, we are diploid, and after meiosis, those cells become haploid. So as I mentioned before, meiosis is a double division. There are two divisions that will take place, and we call them meiosis 1 and meiosis 2. So meiosis 1 is the first mitotic division. There's some very specific and important events that happen in meiosis 1 that don't take place in any other place in this type of sexual reproduction. So the first is your chromosomes have the ability to recombine. And when recombination takes place, there's actually going to be a switching of information or segments between the homologous chromosomes. This is going to increase genetic diversity. Okay, meiosis 1 is going to produce two haploid cells, and those haploid cells are going to have chromatids that are still attached. I'll show you better in the picture that's coming. Meiosis 2, or the second mitotic division, those sister chromatids are actually going to separate from each other, and they're going to end up in separate cells. So in meiosis 1, the homologous pairs separate from each other, and then in meiosis 2, the attached sister chromatids separate from each other. At the end of meiosis 2, we will have made four recombined or recombinant haploid cells. So 4 cells that have some genes that have switched genetic identity, let's say, with each other, and all of the cells will only have half the total number of chromosomes. So the genetics will be similar, but not identical. So again, think back to your siblings. Even if you share your mom and dad with all of your siblings, you guys don't look like each other exactly, and you don't look like your parents exactly. You'll have similarities, but you're not identical. So we sometimes refer to meiosis as reduction division, where we're reducing the number of chromosomes in the daughter cells. Remember, it's always about getting to that half number. So as far as the phases, you will see some familiar steps that we've already talked about in mitosis. So gametic cells are still going to go through interphase. They will still go through G1, S, and G2. And then once they've gone through G2 and they're ready to go through meiosis, they will not go back to interphase until the entire process is done and the 4 haploid cells have been made. So we will have prophase 1, and in prophase 1 is where that recombination, also called crossing over, is going to take place. Then metaphase 1, we'll have anaphase 1, telophase 1, then we have interkinesis, then we'll have prophase 2, metaphase 2, anaphase 2, telophase 2, and another splitting of the cytoplasm for the resulting 4 cells. So if you look at the picture, I'm going to switch to my laser pointer. So this cell has just completed interphase, and it's getting ready to start doing its mitotic division. So note, this cell hasn't actually gone through interphase. This cell is going through interphase. So this cell is diploid because I have 2 of each type of chromosome, but replication hasn't happened yet. I know replication hasn't happened yet because I don't have sister chromatids. Remember, we call them sister chromatids if they are attached to each other in the middle of the chromosome. So replication takes place. I now have sister chromatids, and now I am ready to start the process of meiosis. So in that first meiotic division, that important step of recombination, also known as crossing over, which is the step that's going to add to my genetic diversity, is occurring right here. So if you look at this chromosome, you can tell that it's mostly red and it has a little bit of blue. This chromosome is mostly blue with a little bit of red. These pairs got close enough to each other that they physically switched information with each other. So now mom's chromosome is still mostly mom's genetics with a little bit of dad's genetics that wasn't there before. Dad's chromosome is still mostly dad's genetics with a little bit of mom's genetics that wasn't there before. After the first meiotic division, these pairs will never be together again. So they're going to get separated into one cell, and then this chromosome gets separated into this cell. And that ends meiosis 1. Crossing over happens in prophase 1 of meiosis 1, and it never happens again. So it's important that you remember that. So now each of these cells will go through meiosis 2. They'll go through prophase, metaphase, anaphase, telophase all over again. So let's just use this one as our example. So we go through our various steps, and what's going to happen are the sister chromatids are going to get separated, and they're going to end up in individual cells. Same thing over here. Individual cells. So that's how I go from 2 cells now to 4 cells. Each of these cells will produce 2 new cells. So 2 and 2 for 4. But if you look closely, I have one cell that has 100% mom genes. I have another cell that has 90% mom genes, 10% dad genes. I have another cell that is 90% dad genes, 10% mom's genes, and then I have another cell that is 100% dad's genes. Each of these cells are genetically unique. They are different to each other. Now let's talk about this term of non-disjunction. Non-disjunction can actually occur in 2 separate places in meiosis. Non-disjunction is the failure of the homologous pairs or the failure of the sister chromatids to separate from each other. So in meiosis 1, these homologous pairs should go into 2 different cells. But when non-disjunction happens, both of them end up in one cell, and then the other cell is left empty for that particular chromosome. So that's the first place it can happen. The second place it could happen is when we go through the second set of mitotic divisions. If those sister chromatids don't separate from each other, then we'll end up with one cell that has twice the amount of that particular chromosome, and the other cell will have none of that particular chromosome. Non-disjunction can lead to some different kinds of genetic disorders, which we will cover later on, but the one you might be most familiar with is Down syndrome, which is where an individual has 3 copies of chromosome 21 instead of the 2 copies that he or she should have. Okay, so let's step through the stages of meiosis in a little bit more depth. So again, you're only going to focus on the things that I point out. So in prophase 1, our sister chromatids are going to condense into that chromosome shape. And then we have these 3 new events that take place. So the homologous chromosomes will pair up. This is known as synapsis. When they are fully paired together, they're called tetrads. And because they have crossed arms and legs or linked with each other in tetrad form, when they separate, recombination or crossing over is going to cause a mixture of alleles. Okay, and physically, this is what that looks like. So here's chromosome 1. There's chromosome 2. And if you look at right here and right here, that's what we're referring to as crossing over. Okay, so these are tetrads that are linked together. Okay, then in prometaphase, and remember prometaphase is kind of an in-between step, but this is where things like your nuclear envelopes breaking down, your spindles and kinetochores are starting to attach, very similar to what we talked through in mitosis. Okay, so let's talk about the recombination of these chromatids because it really is key when it comes to genetic diversity. So recombination, like I mentioned, is also called crossing over, and it's a genetic shuffle or exchange that only takes place in prophase 1. That's the only time it can happen. Okay, so those tetrads, those homologous pairs that have properly paired together, are held to each other, and there is an exchange of alleles. Okay, so where they physically touch is known as chiasmata, or some people just call them crossovers, and they are the points at which this exchange is going to happen. So again, let me physically give you a visual for that. So here is one chromosome. Here is another chromosome. Whoops, sorry. Well, you get the point. So this location here and this location here, that's what we refer to as chiasmata or crossovers. Okay, that is the point at which that red chromosome is going to flip-flop information with that black chromosome in those two locations. All right, so here's the picture that the book has, which is probably way nicer than anything I am capable of drawing. So this is a good opportunity for me to point out some vocabulary. So when we talk about a homologous pair, we're talking about the red chromosome and the blue chromosome. They are a homologous pair. When we talk about sister chromatids, we are talking about the left side of the red chromosome and the right side of the red chromosome, or the left side of the blue chromosome and the right side of the blue chromosome. We call them sisters because they are connected. They are attached to each other. Okay? So when crossing over takes place, the two chromatids that are closest to each other, so it's not all the chromatids, but just the two innermost chromatids cross. And in that location, known as a chiasma singular, chiasmata plural, there is an exchange of information. So when they uncross, here is where my red chromosome now has some blue information on it, and my blue chromosome now has some red information on it. This is a good thing. It adds genetic diversity. The more diversity and variety I have, the more options I have for survival, for adaptations, okay? This event is random. Crossing over is a random thing. You cannot control it. And how the parent chromosomes assort themselves is also random. The joining of the sperm and egg, which sperm is going to fertilize which egg, is also random. So all of these things help to increase genetic diversity or variety. And variety increases our ability to survive. If we all were exactly the same genetically, we would all respond exactly the same way to every environmental condition, to every disease, okay? This is going to be gruesome, and I really hope I don't trigger anybody with this, but think back to COVID-19. If everyone was the same genetically, everyone would have responded the same way to COVID-19. Some people caught COVID and died. Some people caught COVID and survived. It's all based on your genetics and the advantages and disadvantages you may or may not have inherited from your parents. All right, so here we are looking at our mitotic division. So we're looking at this first mitotic division. This specifically is showing prophase 1. So here I have those chromatids starting to condense. I am going to start to lose my nuclear envelope. I'm going to start to make spindle fibers. Things are going to start to move. This is where crossing over is going to take place. And at the end of crossing over, I'm going to have some chromosomes that have genetic information that they didn't have before. So that was meiosis 1. So now I'm looking at that was not meiosis 1. That was prophase and prometaphase 1. So now let's continue and talk about metaphase 1 and anaphase 1. And then finally telophase 1. So with metaphase 1, this is very similar to mitosis, except instead of individual chromosomes lining up in the middle of the cell, tetrads, the pairs, are actually going to line up along the metaphase plate. Okay, now that's in metaphase 1. In anaphase 1, those tetrads, those pairs, are going to say goodbye. They're never going to see each other again. They're going to live in different cities, is how I describe it to my high schoolers. So the homologous pairs separate and they move to either side of the cell. Remember, the sister chromatids are still attached to each other. So where one sister goes, the other sister goes. But their boyfriends or girlfriends are not going with them. Okay, think of it this way. Again, this is where non-disjunction can happen, which would create an abnormal number of chromosomes. So instead of having them nicely divided between two sections of the cell, one section of the cell will have more information than it should, and the other section will have none of that particular chromosome. And then in telophase 1, there's no more changes to the chromosomes at this point. Our spindles will disassemble, and we will start to form or reform nuclei around those chromosomes at those either ends of the cell. Okay, so visually, this is what metaphase 1 looks like. So this is what I meant when I said the pairs line up in the middle. So they're not lined up in like a straight line of 4. They're lined up in a double line, if you will. So then in anaphase 1, everybody that's attached to each other goes this way on the red side. Everybody that's attached to each other goes this way on the blue side. And then in telophase, we are going to reform that nucleus. And now our sister chromatids are still attached to each other, but we are no longer in homologous pairs. Now, because we are not about to go back and do replication and any of that again, because we've already done that, interkinesis is the phase in between meiosis 1 and meiosis 2. So straight away we go into prophase 2 and start the process all over again. Okay, so meiosis 2 is made up of prophase 2, prometaphase 2, metaphase 2, anaphase 2, and telophase 2. Important things to note. So just like before, our chromosomes are going to condense, our spindles will form. Our nuclear envelope will break down, and our kinetochores will form and attach themselves to spindles. The chromosomes are going to line up on the metaphase plate, just like they did before. All of this is happening in prophase 1 and in prophase 2 and in metaphase 2. In anaphase 2, the spindles are now going to separate the chromatids. So now the sisters are going to live in different cities. They went to university together, they graduated with their degree, and now one of them's moving to Seattle and the other one's moving to Florida. Okay? Once that happens, in telophase 2, the spindle fiber will disassemble and a new envelope will form around the two new sections that will become the nuclei of those new cells. So visually, this is what this looks like. So remember, this is happening twice because we made 2 cells at the end of meiosis 1. So the cells are going to condense. Here we have our spindles forming. The spindles are moving. They're starting to attach themselves to the chromosomes. Then in metaphase 2, those chromosomes are lined up in that metaphase plate or the equator of the cell. Then the spindles will separate the sister chromatids and they start moving to either end of the cell. That happens in both of them. So that is my anaphase 2. And then finally in telophase 2, we will reform that nuclear membrane and those chromatids will decondense and become long and fibrous-like, spindly-like again. So one of the things that we always do is kind of a side-by-side comparison of meiosis and mitosis, just so we can go over some fine points. So specifically here, we're looking at mitosis. Remember, mitosis is going to produce 2 daughter cells. It has 4 steps. So prophase, metaphase, anaphase, telophase. And the cells produced are going to be genetically identical to the original cell. So in prophase, we are going to condense those chromosomes. They've already been replicated. They are going to line up in the middle in metaphase. The sister chromatids are going to separate from each other in anaphase. And then the nuclear envelope will reform around them. And we will have exactly the same chromosomes. If you look, they're exactly the same. No genetic variation whatsoever. Meiosis, on the other hand, is made up of a double set of divisions, and the goal is not to maintain the chromosome number, but to cut the chromosome number in half. So in prophase 1, the very first, not the very first thing, but one of the most important things that happens is recombination or crossing over, where homologous chromosomes exchange genetic information with each other randomly and spontaneously. Once that happens, those homologous pairs are going to line up in the middle of the cell for metaphase 1. The homologous pairs are going to separate themselves in anaphase 1. And then in telophase 1, we have homologous pairs. Sorry, we no longer have homologous pairs. We now just have sister chromatids that are attached to each other. And we have made 2 cells. So from this point on, each of those cells will go through prophase 2, where, again, they attach themselves to spindles. No homologous pairs this time. Then in metaphase 2, they will line up individually along the middle of the plate. And in anaphase 2, the sister chromatids separate from each other and they move to opposite sides of the cell. And then in telophase, we are going to surround those sister chromatids with nuclear envelope, and then the cells themselves will split in 2, creating 4 genetically unique and different cells. Also note the cells only have half the information of the original parent cell. And that's it for meiosis. Bye, guys.